Hello friends, today I will be talking about an overview of gene regulation. Regulation is about decision making. For living organism, it is how cells control when and how much of its genes or a set of genes are expressed. Since the topic gene regulation is both very deep and broad, today I will give you an overview of it. Now the question is, what is gene regulation? And the definition says it is a fundamental process that cells use to control the timing, location, and amount of gene expression. So, the word fundamental here means primary mechanism that sustains life. And to ensure this, the particular gene is expressed at particular time and amount, the genes are either switched on or off. And the discovery of gene regulation system is widely considered as Lac-Operon, and it was discovered by Francois Jacob and Jacques Monod in 1961. Now here we will address the question why gene regulation is important. So these are the critical roles of gene regulation, and it includes its role in cellular differentiation, in energy and resource conservation, in environmental adaptability, and in disease prevention. So we know that every cell in the body of an organism shares same DNA. Now gene regulation determines whether the cell will be differentiated into a neuron or muscle cells or any other type of cells. Now coming to the second point, that is its role in energy and resource conservation. A cell utilizes significant amount of energy and resources to produce a protein. And gene regulation ensures cells only synthesize the protein that are actually needed at any given moment. So switching on or switching off the gene as per the need helps the cells to save energy and resources. Now coming to the fourth point, that is its role in environmental adaptability. Gene regulation helps organism to quickly respond and adapt to the external factors such as temperature fluctuation or toxins or if there are some changes in nutrient availability. And the fourth point is the role of gene regulation in disease prevention. Here I can say that gene regulation prevents disease by acting as a biological quality control system that stops damaged cells from multiplying. When gene regulation functions correctly, it activates protective gene to repair cellular damage, and if gene regulation fails, mutated cells can grow, which may lead to life-threatening disease like cancer. The classic example of gene regulation in preventing disease is the p53 gene, which we all know is the guardian of the genome. Its primary role is to control the cell division and maintain the genetic stability. Now coming to the slide which shows prokaryotic versus eukaryotic gene regulation. And the difference arises due to the difference in the cell structure. Here in prokaryotes, nucleus is absent, so DNA is found in the cytoplasm. Whereas in eukaryotes, nucleus is present, so DNA is found within the nucleus. Here in prokaryotes, transcription and translation occurs almost simultaneously, whereas in case of eukaryotic organism, transcription occurs in nucleus, whereas translation occurs in cytoplasm. So we can say that in prokaryotes, gene regulation is primarily at the transcriptional level, whereas in case of eukaryotes, the regulation takes place at many levels because of the structural complexity. Now coming to gene regulation in prokaryotes. The DNA of prokaryotes is organized into a circular chromosome supercoiled in the nucleoid region of cell cytoplasm. The figure here shows a DNA segment which has a regulatory gene and an operon. Regulatory gene encodes for repressor protein, and operons are a cluster of functionally related gene controlled by a single promoter and operator. Promoter here acts as landing pad where RNA polymerase binds to and transcription takes place. Operator, which is present between the promoter and structural genes, is we can say it as a switch or a traffic signal where the repressor protein comes and binds and blocks the function of RNA polymerase. So the three regulatory molecules in gene regulation prokaryotes are activators, repressors, and inducers. Activators binds near the promoter and enhances the function of RNA polymerase. Repressors binds to the operator and blocks RNA polymerase activity, and inducers binds to the repressors and inactivates it. Examples of gene regulation in prokaryotes are lac-operon and tryptophan operon. Taking the example of lac-operon, so this is the DNA segment which shows the regulatory region and the lac-operon region. In the regulatory region, it is promoter for repressor gene, and lac-operon consists of promoter for lac-operon, operator, and the structural genes. Structural genes here are Z, Y, and A. Z encodes for beta-galactosidase, Y for permease, and A for transacetylase. So these are the enzymes used in lactose metabolism. So what happens when there is no lactose in the environment? Repressor protein is formed which goes and binds to the operator, and so this when repressor binds to the operator, it prevents the transcription of the structural genes, that is Z, Y, and A. And the second one here, it shows that when lactose is present in the medium, the isomer of lactose, that is allolactose, goes and binds to the repressor protein, which causes the conformational change in the complex, so that the repressor protein fails to bind to the operator, and thus the transcription of the structural genes takes place. Now coming to gene regulation in eukaryotes. Because of its complex cellular structure, eukaryotic gene regulation is highly complex, multi-step process by which cells control when, where, and how much of a specific protein is produced. Various levels at which gene regulation takes place are at the epigenetic level, transcriptional level, post-transcriptional level, translational level, and post-translational level. So the first three takes place in the nucleus, and translational and post-translational level takes place in the cytoplasm. Now coming to the epigenetic level. Here changes in the regulation tends to bring changes in chromosomal structure, which leads to the genes to be turned on or off. And the regulation occurs by the chromatin modification like DNA methylation or histone acetylation, etc. Now the second one is the transcriptional level. This is the primary and most strictly controlled point of regulation. Here the cell decides whether mRNA will be synthesized at all. And so here it requires a combination of transcription factors, enhancers, and silencers. The third level is at the post-transcriptional level or RNA processing. Once the pre-mRNA is transcribed in the nucleus, it undergoes changes before moving into the cytoplasm. So here splicing, RNA capping, and addition of poly-A tail takes place. Now the fourth regulation, which now comes into the cytoplasm, is the translational level. So here what happens, once the mRNA is formed and it has been transported to the cytoplasm, now the fate of mRNA is decided whether it will go for the protein synthesis or it will go for the degradation, or depending on the cellular need. So here we can say that for degradation or downregulation of the mRNA, miRNAs come into play and they block translation or trigger their degradation by RNA interference. And the last one is the post-translational level. After the protein is synthesized, its activity can be altered by addition or removal of chemical groups like phosphorylation, which affects the function of the protein. And also if the protein in this level, the post-translational level, protein can also be tagged with another protein such as ubiquitin, which marks it for the destruction. This is one of the examples of gene regulation in eukaryotes. The figure shows the p53 signaling pathway. So in normal condition, what happens? P53 is maintained at a very low level, and P53 is downregulated by MDM2, MDMX complex. MDM2 is E3 ubiquitin ligase. MDMX suppresses the transcriptional activity of P53. So this complex together uh, stimulates the degradation of P53 through ubiquitination pathway. So what happens when the cell is in stress? So in the stressed condition, P53 responds to a range of environmental and intracellular stresses, including agents that cause DNA damage, UV radiation, hypoxia, or oxidative stress. So as soon as the stress is there, several kinases get activated such as ATM ATR, which brings the conformational changes in P53 MDM2, and this causes the blocking of their interaction and resulting in the stabilization of P53. And once the P53 is stabilized, it acts as transcription factor to turn on the specific target genes. And the outcome depends on the extent of damage. So if there is if the cell needs the DNA to be repaired, the transactivation takes place of PCNA GAT45, and for the cell cycle P50 arrest, G1S cell cycle arrest, P53 trans activates P21WF1, and if there is the cell is severely damaged, the pro-apoptotic gene like BAX is activated and cell goes for apoptosis. So this shows how P53 signaling pathway determines the fate of the cell depending on the damage caused to it. So today I was able to give you a brief idea about gene regulation, its definition, importance, and its role in prokaryotic, how the gene regulation takes place in prokaryotic and eukaryotic cell. So gene regulation is a very vast topic, and I can say that it bridges the gap between the genetic potential and physical reality. Gene regulation is the master conductor of cellular life. It ensures that every cell in an organism contains the exact same genetic blueprint, yet possesses the distinct identity and function required for the survival. Thank you.